All right, ready to dive deep. Today, it's all about FACS analysis.
FACS analysis.
Yeah, it's this super cool way scientists study cells.
Oh, right. Cells.
You know, like the tiny building blocks that make up, well, everything.
Right.
It's crucial stuff, especially for things like researching diseases and developing new medicines. But there's a bit of a catch. Sometimes these cells, they try to trick the scientists.
Trick them. How?
Think cellular disguises.
Okay, now you have to explain.
So picture this. Two cells get all buddy-buddy, stick together, and pretend to be one big cell.
Oh, I see. So they're kind of like,
Exactly. Scientists call them doublets.
doublets. Sneaky. But why are these doublets such a big problem for scientists?
Well, imagine you're trying to count people in a crowded room.
Okay, got it.
But some are holding hands, so you think it's one giant person.
Makes sense. Your count would be totally off.
And it's the same with cells. Doublets can make it look like there are fewer cells or they're bigger than they really are. And that messes up all sorts of important measurements.
Right, right. I can see how that'd be a problem. But how do scientists even know these doublets are there? I mean, cells are tiny.
It's like a microscopic game of hide and seek, right?
Totally.
But scientists have this amazing tool called a FACS machine.
FACS machine, what's that?
It shines a laser on the cells and measures how the light bounces off.
A laser? For cells, that's pretty high tech.
It is. So tell me, what can scientists learn just by looking at how the laser light bounces off these tiny cells?
Well, for one, they can figure out the cell's size. Think of it like shining a flashlight on something.
Okay.
The bigger the object, the bigger the shadow it casts.
Right, that makes sense.
FACS basically measures that shadow to figure out the size of the cell.
So if you have a doublet, which is like two cells stuck together, it would cast a bigger shadow.
Exactly. It's a dead giveaway.
Wow.
But that's not all.
Oh, there's more.
Oh yeah, FACS can also tell you about the graininess of a cell.
Graininess. What's that?
Think of it like the texture of a surface, like how rough or smooth something feels. FACS can tell how much light is scattered off to the side, kind of like how a rough surface scatters more light than a smooth one.
Interesting. So a rough and bumpy cell would scatter more light.
Precisely. And guess what? Doublets, being a bit clumpy, tend to scatter more light than single cells.
Ah, so by looking at the size and the graininess, scientists can start picking out these sneaky doublets.
Exactly. It's like those spot the difference puzzles, but with cells.
I love that analogy. So they're using lasers, shadows, and even textures. What other clues help scientists catch these doublets?
Well, get this. FACS machines are even more sophisticated than that. They don't just look at the overall size and graininess, they actually measure the cell's area, height, and width as it passes by the laser.
Hold on. Area, height, and width?
Okay.
It's hard to picture measuring something that small in 3 dimensions.
Right. It's pretty amazing. Okay, picture this. Imagine shining your flashlight on a cell again.
Okay, got it.
The area is like the overall shape of the shadow it casts. The height is like measuring how tall the cell is at its tallest point. And the width is like measuring how long it takes the cell to completely pass through the beam of light.
Wow, okay, so they're basically measuring how long it takes for the cell to cross the laser beam's path.
That's a great way to put it. And here's why those measurements are so important for figuring out which cells are doublets. Doublets are larger, obviously, and potentially oddly shaped, so they'll have different area, height, and width measurements than single cells.
Oh, I see. It's like if you tried to fit a square peg in a round hole.
Yeah.
The doublets just don't measure up the same way as the single cells.
Exactly. And scientists use that to their advantage. They can set up these special gates on the FACS machine to filter out cells based on these measurements. It's like,
Wait, gates?
yeah, think of them like checkpoints that the cells have to pass through.
I get it.
If a cell's measurements don't match up with what a single cell should look like, then it gets flagged as a potential doublet, kind of like being sent to timeout.
So we've got these sneaky doublets trying to blend in. And scientists are using these high-tech gates to sort them out. You mentioned there's a step-by-step guide in the article for this whole gating process. Can you walk me through how that works?
Of course. It's basically a series of steps, each one designed to catch a different kind of doublet.
So like a multi-level security system.
Exactly. First, we use something called forward scatter to get rid of the really obvious ones, the big doublets.
That's the first line of defense, right? Catching the most obvious rule breakers.
You got it. But some doublets can still slip through. So we use another gate that looks at the width of the light signal. Remember how we were talking about how doublets tend to produce wider pulses of light because they take longer to pass through the laser beam?
Right, right. It's like using a finer mesh net to catch the smaller fish.
Exactly. We can even take it a step further by looking at the side scatter, which, remember, tells us about how complex the cell is on the inside.
Right, like adding another set of fingerprints to the analysis, so we don't accidentally lump different types of cells together.
Exactly. And if the cells are labeled with these fluorescent markers.
Oh, right. You mentioned fluorescence earlier. Now you have to tell me more. We're talking like glowing cells here.
We are. Scientists can tag different types of cells with special markers that glow when they're hit by the laser, kind of like a color code.
Oh, that's cool. So each cell type gets its own color.
Yep. But here's the thing about doublets. Being a mix of two cells, they can sometimes have mixed up colors, and that can really mess things up.
Ah, so they're wearing two different jackets, and the scientists need to make sure they're not fooled by the wardrobe malfunction.
Exactly. That's where another gate comes in. We use it to make sure that the fluorescent signals make sense and are coming from single cells, not mixed up ones.
It's like a whole detective story, using all these different clues to find the culprits.
Hmm.
I'm really starting to get why getting rid of these doublets is so important. But is it always this complicated? Does it ever get easier?
Well, every experiment is different, so there's no one-size-fits-all approach. Scientists have to figure out which gates to use based on what cells they're working with.
So it's not a set it and forget it type of thing.
Definitely not. There's a lot of fine-tuning involved. And to make sure they're doing it right, scientists often use what are called control samples.
Control samples, what are those?
Think of them like the known knowns. These are samples where we're absolutely sure that all the cells are single cells. By running them through the FACS machine, we can make sure that all the settings are right and that we're correctly identifying the doublets.
Ah, so it's like a test run to make sure everything is calibrated.
Exactly. It really shows just how careful scientists have to be when it comes to this kind of analysis. Even something as seemingly small as doublets can really impact the results, so getting it right is crucial.
You know, listening to all of this, I'm realizing that this is way more than just a technical detail. It's about making sure the data scientists get is reliable so they can draw the right conclusions. But why does all of this matter in the grand scheme of things? What's the real-world impact of getting this right? We'll explore that and more in part 2. Welcome back. So we've been talking about all these sneaky doublets, right? And how scientists are like detectives trying to track them down in their FACS analysis.
Yeah, exactly. It's like a whole cellular whodunit.
I love that. But I'm curious, why does all of this meticulous work even matter? Like in the grand scheme of things, what are the stakes of getting this right?
It's a great question. You see, it's not just about being super precise in the lab. It's about making sure we're getting the whole picture and can make good decisions, especially when we're talking about things like health and medicine.
Okay, so can you give me an example? How could something like doublets actually affect people's lives?
Okay, well, imagine researchers are testing a new cancer drug. They're using FACS analysis to see how the drug affects different cells in a tumor.
Makes sense.
But what if those doublets aren't properly filtered out? The data could end up being totally misleading.
Oh, wow. How so?
Well, let's say the drug is actually working great, killing off cancer cells left and right.
Okay.
But because of those doublets pretending to be single cells, the data might show that fewer cells are being killed than there actually are.
So it's like the doublets are messing up the numbers, making the drug look less effective than it really is.
Exactly. They're throwing off the whole count, and that could mean that a good treatment gets ignored or even abandoned.
That's a scary thought.
It is, isn't it? And you know, the opposite could happen too. If a drug isn't actually working well, those doublets could make it look more effective than it really is.
So it's a double-edged sword.
Exactly. And that could give patients false hope, or even worse, lead to harmful side effects. Getting the cell analysis right is crucial for making sure we make the right choices about treatment and aren't misled by these doublets.
That's a pretty sobering thought. It really makes you see the importance of all of this. It's not just about some numbers on a graph. It's about real people's lives.
Exactly. And it goes way beyond just drug development. I mean, getting the cell analysis right is important for tons of different areas of research, from the immune system and infectious diseases to things like stem cell therapies.
So it's all about getting the story straight at the cellular level, because these tiny cells, they hold so much information about how our bodies work, how diseases happen, and how we might be able to treat them.
Precisely. And that's why scientists are always trying to improve things like doublet discrimination. They're working on new algorithms, better software, and even new FACS machines that can that can do even more.
It's like they're constantly upping their game.
Right. They're always trying to stay one step ahead of those tricky doublets and get the best data possible.
It reminds me of those spy movies where they're always coming up with new gadgets and techniques.
That's a great analogy. So, knowing just how crucial accurate cell analysis is, what do you think the future holds? What other innovations could be on the horizon to make things even better? Like, what's next in the world of cellular detective work?
That's a good question. What do you think?
Well, one thing that has a lot of potential is the use of artificial intelligence and machine learning.
AI for cell analysis.
Yeah. Imagine having software that could automatically identify and get rid of those doublets even better than we can do now.
Wow, that would be amazing. Like having a superpowered assistant.
Exactly. It would not only save researchers a ton of time, but also really reduce the possibility of human error.
So it's like taking the human element out of the equation, making the whole process faster, more efficient, and even more accurate.
Precisely. And that's not all. Scientists are also creating these incredible new fluorescent markers.
Markers.
Yeah, the things that make the cells glow. They're making them even brighter and more specific, which means they can tell different cell types apart even better.
Hold on, even brighter colors. What's the advantage there?
Well, the brighter the color, the easier it is to see it, right? It makes it much easier to tell different colors apart. And with these new markers, scientists can track specific cell populations with amazing precision and sensitivity.
So it's like upgrading from a basic flashlight to a high-powered spotlight.
Exactly. It's like giving scientists a stronger magnifying glass, you know? It allows them to see even more detail in this incredible world of cells.
It's amazing what scientists can see these days, and it sounds like things are only going to get more impressive. But before we get too caught up in the future, I think we should take a moment to appreciate how clever the scientists who developed all of these techniques in the first place were.
Oh, absolutely. FACS analysis and doublet discrimination are, well, they're proof of just how curious humans are and how much we want to understand the world. It's amazing to think that even the tiniest details can have such a big impact on how we understand things and how we can improve human health.
That's a great point. It's easy to get lost in the technical details, but at the end of the day, it's about pushing the boundaries of what we know and then using that knowledge to make things better.
Exactly. And learning about all of this has really opened my eyes to the amazing world of cells and the incredible work that scientists are doing.
Me too. It's just, I don't know, it's pretty inspiring.
It is. And the more people who understand and appreciate all the intricacies of scientific research, the better. It means we'll be in a better position to support it and benefit from its discoveries.
Well said. We've covered so much today, from how FACS analysis actually works to the broader implications for scientific discovery in human health. But there's one more aspect of this topic that we need to discuss, and that's the question of ethics.
Oh, right, of course.
As we get better and better at analyzing and manipulating cells at such a fine level, we're definitely entering uncharted territory.
You're right, we are.
So stay tuned for the final part of our deep dive, where we'll tackle those crucial ethical considerations.
Welcome back to our deep dive. We've been on quite a journey, from lasers and shadows to doublets and cellular detective work.
It really has been a fascinating exploration.
It has, but before we wrap up, we need to talk about something that's been, well, kind of lurking in the background.
Oh, you mean the ethics of it all?
Exactly. As we gain more and more power to analyze and manipulate cells, with such incredible precision, we're stepping into some uncharted territory.
You're absolutely right. It's like that old saying, with great power comes great responsibility. As our understanding of cells grows and our ability to actually manipulate them advances, we need to be really careful that we're using this knowledge ethically.
Okay, so where do we even begin to unpack the ethical dimensions of something like, well, FACS analysis? I mean, it seems pretty technical.
Right. It does, but it really does connect to some bigger questions about how we use science and technology. I think a good place to start is with the data itself.
The data. Yeah. We've talked about how important it is to have accurate data, without those doublets messing things up. But even with perfect data, there's the question of interpretation. How do we make sure the information we get from FACS analysis is used responsibly, ethically?
Right. It's like Having all the pieces of a puzzle but not knowing how to put them together or what the picture is even supposed to be.
Exactly. And it gets even more complex when we start thinking about the different ways FACS analysis could be used. Like, take personalized medicine, for example.
Oh yeah, using FACS to figure out how someone might respond to a specific treatment.
Precisely. But what if this information is used to, I don't know, discriminate against certain patients? Or what if these personalized treatments are only available to people who can afford them? These are some of the ethical dilemmas we have to think about.
It seems like we need a whole new set of rules, a whole new ethical framework to keep up with all of these scientific advancements.
I think you're absolutely right. As our knowledge and capabilities expand, we need to have these ongoing conversations about the potential societal and ethical implications. It's not just about what we can do, but what we should do.
You know, it's kind of mind-blowing to think about how far we've come from trying to figure out what a doublet is to grappling with these massive ethical questions
Yeah.
about how we use scientific knowledge. It makes you realize science isn't just about facts and figures. It's about our values and how we use discoveries to shape the future.
That's a really powerful insight, and it highlights why it's so important to have these conversations, and not just within the scientific community, but with everyone. I mean, we all have a stake in how these technologies are developed and used.
Absolutely. It's not just up to the scientists to figure this out. It's a collective responsibility. We all need to make sure these powerful tools are used ethically and, for everyone's benefit.
Well said. It feels like we've gone pretty deep today. We started with what's a doublet, and ended up exploring the frontiers of science, ethics, and the future.
It's been an amazing journey. And I think it's safe to say we've all learned something new about FACS analysis, cellular detective work, and the importance of, well, responsible innovation.
I couldn't agree more. I hope everyone listening has a new appreciation for the complexity and wonder of the cellular world, and for how important ethical considerations are, especially as we move forward with science and technology.
That's a perfect way to wrap things up. Thanks for joining us on this incredible journey. Be sure to check out the show notes for more information and visuals. And until next time, stay curious.